Boa tarde a todos. É o meu prazer estar aqui hoje nos webinars do InfoBlender. Hoje vamos ter uma talk do Vinicius, que é investigador no LASIG, da Universidade de Lisboa. Está agora a acabar o doutoramento, esperamos muito brevemente que possa defendê-lo, e tem trabalhado nas áreas de sistemas distribuídos, sistemas de armazenamento. E hoje esta apresentação é sobre um artigo que foi publicado na I3S Transactions on Computers. O artigo é sobre como é que podemos otimizar o armazenamento de dados genómicos e como é que podemos tornar este armazenamento mais eficiente em termos de não só da velocidade de acesso aos dados, mas também em termos de custos de armazenamento. E por fim este trabalho resulta de uma colaboração entre o LASIG e o ASLab, daí também ser algo interessante para apresentar nestes webinars. Pronto, sendo assim, Vinicius,
Ok,
então,
passar para o inglês, já que a apresentação estava em inglês, já fica mais fácil. So thank you, João, for introducing me and for inviting for this talk. It's a pleasure to be here today, and it was a pleasure to collaborate in this work with you. Hope this is the first from many to come. And as Ron said, this is a collaboration between LASIG and HasLab, because HasLab has contributed previously with a series of deduplication articles and studies and research. So this was a topic that we are not very used to. So we invited João Paulo to work with us, and it was really a really interesting collaboration. I would like also to thank Paulo Rodrigues for organizing and promoting the webinar, and also for you that are here today watching this. So to start, this work is the GenoDedup is a similarity-based deduplication Delta-Encoding for genome sequence data. And I will start with a brief introduction regarding human genomics data. So basically, human genomics is a multidisciplinary area that has been advanced in many critical areas that are increasing our health awareness and life expectancy. One interesting thing is that accessing large data sets of genomes is a long-term ambition from both clinical and research communities. So both of them would like to have access to large sets of human genomes to reach to novel medical breakthroughs that increase even further our health awareness and life expectancy. So the main players in this game are biobanks and hospitals, because they already store or stock thousands to millions of physical biological samples. So these samples like blood or tissue and many other samples of interest. But now they are also under pressure to start storing the digitized version of this sample. So they have to sequence these genomes and they have to store that. However, this is not their expertise. So, and they have also a limited budget that they have to invest in reagents and chemicals to sequence the data and so on. This budget is limited, and they having to spend it on storage servers and processing servers is a little bit more complicated. And even it's not their expertise. So it's common to not seeing they storing efficiently this type of data sets. Just to have an idea of the size of the problem size, I have here one study from Berkeley University from 2012, where they estimated the cost to store 1 million human genomes. How much would cost it? So they assume usually one human genome, or the data from one cell, it's almost 300 gigabytes. So they assume that each genome is 165 gigabytes. They assume some sort of compression. And for 1 million genomes, it gives us 165 petabytes. So their estimation cost is $65 million for storing and processing. It's not only storing, but during the first year. So it would be the capital expenditures. to create a private infrastructure for storing this 1 million of genomes. And then after, on each subsequent year, they would have to spend another 35 million to upgrade this and keep this infrastructure updated, and also working with electricity and everything else they need. So it's a lot of money that if you can imagine, for storing a million, any hospital or any biobank has more than 1 million samples, or it's common to have more than that. So imagine having each one of them having to spend this amount of money to create only a storage and processing infrastructure. So it's a lot of money. And even if you consider, okay, let's put everything on a public cloud like Amazon. I calculated here pretty quickly. The cost to store this 165 petabytes on cold storage on Glacier, which is $7.9 million per year, or in an infrequent like S3 access, infrequent access storage is almost $20 million per year still. So it's a lot of money, and we have to. Our goal with this work was to try to balance the storage space and the restore performance. So this is our goal, trying to balance these two components. So the outline of this presentation, I divided in other six topics. I will first explain what are the genome files we are talking about. I will present 3 studies that are within this paper, which is about compression, the identity-based deduplication, similarity-based deduplication, and the final remarks. So regarding the genome files, it basically starts with a sample donor that provides the biological sample. It is sent to a data sequencer, and this data sequencer will sequence the sample and generate a genome file. There are 3 main data representations for genomic data. The first one is the sequence data, which is the raw data coming from sequence machines. Then this data does not come as a single contiguous genome. So it's a lot of small chunks of DNA that come unaligned. You don't know which portion is sequenced at each part of the file. So there is a reference genome that is used to align these entries into this reference, and then you create a new version of file, which is the aligned genome, the second line here. And then there is the third one, which is just merging these alignments and then create a contiguous human genome. So what we are interested here is in the first case, because this is the raw data coming from the sequencing machines. This is the hardest data to store. And I will present a few more details about that. So imagine a text file, a huge text file with 300 gigabytes, composed of entries. For instance, 1 billion of entries. Each entry is composed of 4 lines. So we have 2 comment lines, and we have the DNA that was sequenced by the machine, and then the 4th line is the quality score. So these quality scores are some scores to attest how sure the machine is on sequencing that DNA. So for each nucleobase sequenced by the machine states which is the confidence level it has on that nucleobase. So this is an aligned data, as I mentioned. And then the comment lines here, they have more or less a fixed structure. They have some identifiers of the sample, the identifiers of the entry, and so on. It is easy to compress this data. There is this algorithm from Bola et al., where they compress it more or less 17 times. So it's okay. We are not focusing that in this work. So we use their work to compress the comment lines. Then DNA is a little bit easier to compress because we have the human genome reference. So we can take each one of these lines, find its position in the reference and say, okay, is that position plus this or that modification. So it's easier to compress, mostly because 99.9% of these letters match exactly with the human reference. So it's easier to create what they call the referential compression. And also one thing that is easy is to compress the DNA is because the alphabet is only 5 letters. So it's a small alphabet to create a compression encoding, for instance. So the main problem of these files are in the quality scores. So this is the hardest part to compress because there is no reference. The first part is that. There is no way to align one line of this in a big file. And the alphabet is way, way bigger. So now we have 93 options in this alphabet. So the first contribution of this paper is that we analyzed several patterns or aspects from quality scores that matter for the duplication. So we noticed, for instance, that and there are most of these patterns have been identified. We only centralized them. For instance, only 40 of these levels of confidence are actually used. The other 53 are not used yet because the machines have not that much confidence. And the bigger the sequence we are sequencing here, the bigger is the uncertainty at the end. So if we can see at the end of this line, there are lots of hashtags which represent the 0 confidence level. Other pattern that we found is that subsequent quality scores vary little. So from one quality score to the next one, the variance, how much this value varied is very little. So we can create delta values that will convert this type of data to a normal distribution, because most of the case will be the value 0, -1, 1, and so on. So it becomes a normal distribution. And this is the second contribution from this paper. The first contribution of this paper is that we propose to create the circular deltas. So we convert these values using modulo operand, and it reduces the encoding size with the normal distribution. This is just a detail that I'm not entering too much details here. So the evaluation data set that we use in the remaining of this presentation is are 5 files, FASTQ files from the 1000 Genomes project. They sum up 265 gigabytes of data and more than 1 billion FASTQ entries. So regarding the compression of this data of these files, we explored more than 20 compression tools and selected the best 4 generic compression tools and the best 6 specialized tools based on the compression rate and the read-write throughput. These are the explored tools, and the green are the generic selected ones, and the yellow the specialized selected ones. All of them explore intra-file similarity, so they don't use 2 genomes to compare with each other and compress them, except for those using referential compression, where they have the human reference and compress using only that part. So it's a limited inter-file similarity, so it's not even can be considered inter-file similarity. We also discarded lossy compression tools because hospitals and biobanks, they spend a lot of money to sequence this data, so they don't want to lose this data. So they want to have exactly store and process exactly the original data coming from the sequencing machines. So just to have an idea of the results, we have here that Spring is the best tool that compresses more. So it reached to 6 times of compression ratio on average. We have the DSRC, which is the fastest one to write files with more than 1 gigabyte per second. And we have the SecDB, which is the fastest one to read this data, which reads at 127 megabytes per second. So we have also the PIGS, which is the parallel version of the gzip, and the ZPEC, which is the generic compressor that reaches the best compression rate. So with these results, our first question was to check is it an opportunity for the duplication? So can we explore interfile similarity? And also, this is one of the insights that João gave to us. Okay, the duplication usually reaches a high restore performance. So why don't you focus on that as well? So this is something that we started to exploring. Try to check if there is an interface similarity that you can explore with the duplication and obtain the high performance from the duplication. So we did an experiment with identity-based deduplication to show if it is possible or not to obtain the better results with that. So the idea is to find exact duplicates. So we divided in 3 types. But as a spoiler, the conclusion was that all of them fail on FastQ. So I'm going to pass really fast on each one of them. The first one is the file deduplication. So take the file, the whole file, calculate the content hash, and compare the content hash of all files. So we analyzed the 1,000 genomics projects. They have more than 200,000 FastQ files, and less than 0.007% are duplicates. The problem is that each institution has their own samples IDs. Even sequencing the same sample results in different FastQ files. And FastQ is big, so it does not, it discourages having duplicates in the system. So it's spending storage space. So this is not interesting. The second one is block deduplication. So we split the FastQ files in blocks in several sizes, fixed and variable size blocks. And also from the 40 million fixed blocks and 23 million variable blocks that we divided these files, there are non-duplicates. So the idea is that having it's very unlikely to have duplicate blocks because they have the unique sample identifiers and the entry identifiers. There are mutations on DNA and they are sequenced in no specific order. So changing a little bit the order of the sequence will change completely the FastQ file. And the QS, the quality scores distribution varies from run to run. So sequence 2 times the same sample will result in different quality score distribution. So finally, the third one was application-aware deduplication, which considers the file structure. So we know that each 4 lines compose an entry. So we deduplicate this line type separately, and comment lines can be parsed internally. So from the 471 million entries in the DNA lines, 44 million of them are duplicates. So it's equivalent to 9.4% more or less. And the quality scores, only 468,000 lines are duplicates. So it's less than 0.01%. So it's really low. And why we consider it fails? First, human DNA sequence similarity is above 99.9%. And here we have only 9.42%. It's 10 times less what is expected. And also there are studies that show that it's worth to deduplicate spatial data if it reaches 20 to 40% of duplicates, and none of them have reached. So the thing is that now we have an opportunity to explore similarity-based deduplication. So our idea here was to separate the FastQ lines like the last case, the application specific, and to find the most similar entry in a deduplication index. After that, we create a pointer to this similar entry and store the pointer plus the list of modifications to this. One interesting thing is that having only one entry is enough to deduplicate 100%, but the list of modifications can be huge, so it's not interesting, but it's possible. But as we grow the number of the duplication candidates or entries in the index, there will be less modifications, and then we start compressing more. So there are three main aspects three challenges that we have to face here. I will not enter in too much detail here, but basically find a distance metric and encoding. We have Hamming, Leverstein, and we have proposed in this paper another one, Delta Hamming. This is for those that already work with this area. They are pretty simple. Then we have to model the deduplication index. So we applied a clustering algorithm based on previously seen genomes to create a generic deduplication index. So we used the bisecting k-means. We implemented that on Apache Spark and created the duplication index from 1 to 1 million entries. However, we could not, in our experimental environment, we could not run this algorithm with more than 1 million clustering, with more than 1 million clusters. So we could not run that. So it's an opportunity for work in the future. And lastly, we also implemented a locality sensitivity hash, which basically divides the duplication index in several buckets. And when you query one entry, one line, it will say, okay, is on these buckets are the most similar entries to the queried one. So you only perform the optimal search within each bucket. So it reduces a lot the number of comparisons that you need to do. So basically, one example is that from 1 billion elements, you reach to you have to compare only 30 with 30 each query. So it reduces a lot. And then the encoding is the modification to the best neighbor found within this bucket. So this is basically our workflow. Our algorithm receives the FASTQ entries, separates the file types, and then queries the LSH index, which then is used also a key value star to retrieve the values. And then we compare all the entries in that bucket to decide which is the nearest neighbor, and then to create the list of modifications for that. So we start this file. This file is then read by a client, the duplicated one, which reads only the key value star to recover only those selected best neighbors to recover the original file. So our experimental evaluation has resulted in the following. Our compression rate have reached separately on each type of data. Our comments, we used the Bola et al., which is 17.26 times of compression. The DNA with our algorithm, we have reached with the human reference genome and our Delta Hamming, we have reached 13.42 times of compression. And quality scores with only 256 208 quality scores entries candidates, we have reached to 1.88 times of reduction. So summing up everything, we reached to 4.09 times of compression with a write throughput of 0.3 megabytes per second, which is the throughput of the sequencing machine. So we stopped it there at this point because it was enough. And the read throughput, which is our focus, is 208 megabytes per second using 24 threads. So just to compare that, we have just to present some improvements, we can have bigger indexes result in better compression rate in the quality scores. For instance, here from 1.88, we go to 2.46 going to 1 million entries. And then it results in going from 4.09 to 4.92 times of compression rate in total. Our write throughput can be improved using less entries to compare, if the SH is configured to return less entries. And also we can use multi-thread to improve that in the future. So just to compare, ZPack is the generic compressor with the best compression rate in general. So we compressed each type of data separately, and it results in these results. They are only better on this, on the quality scores, but we already reach almost 80% of their result. And also, if you look, their read throughput is way slower than ours. So this is an interesting trade-off. We reach 80% of their results in compression, but then we are 200 times faster than them for reading data. So another experimental evaluation comparing those results from the first table, we have that in the right, we are still slower than all of the others, but we can prove it with some suggestions we have in the paper. But our read performance is way faster, and we have balanced the compression ratio from specialized algorithms to between specialized algorithms and generic algorithms. So just to have an idea of the impact of this, if we have the 1000 genomes project, they have more or less the equivalent to 370 terabytes of FastQ data and compress it. They store this data using gzip in 115 terabytes of compressed data. With our approach, they would have reached to 22% of gains in storage space, and also their ecosystem would benefit from a faster restore because we are 5 times faster than gzip. Just as the final remarks, FastQ compression has its limitations. Identity-based deduplication fails on FastQ. We proved that on the paper. And it opens an opportunity for similarity-based deduplication. So our challenges that we addressed here were to model the duplication candidates, to have large deduplication indexes. We tried to identify efficient search algorithms and the best similarity distance and encoding. We balanced the store space and restore performance by achieving 67.8% of the reduction from Spring. And we restore 1.62 times faster than SecDB, which is the fastest one competitor, a specialized algorithm for FastQ compression. As future work, we intend to improve the throughput with multithread and try to find ways to model bigger deduplication index, for instance, to reach 1 billion entries from billions of examples, previously seen example. And we intend to publish a more complete LSH implementation that enables using this 1 billion entries to deduplicate the human genomes. So this is the link for the article if you have any further thoughts there. And so thank you.